In the audience was Seymour Benzer, yet another ex-physicist who had heeded the clarion call of Schrödinger's book. He immediately understood what our breakthrough meant for his studies of mutations in viruses. He realized that he could now do for a short stretch of bacteriophage DNA what Morgan's boys had done 40 years earlier for fruit fly chromosomes. He would map mutations, determine their order, along a gene, just as the fruit fly pioneers had mapped genes along a chromosome. Like Morgan, Benzer would have to depend on recombination to generate new genetic combinations. But whereas Morgan had the advantage of a ready mechanism of recombination, the production of sex cells in a fruit fly, Benzer had to induce recombination by simultaneously infecting a single bacterial host cell with two different strains of bacteriophage, which differed by one or more mutations in the region of interest. Within the bacterial cell, recombination, the exchange of segments of molecules, would occasionally occur between the different viral DNA molecules, producing new permutations of mutations, so-called recombinants. Within a single astonishingly productive year in his Purdue University lab, Benzer produced a map of a single bacteriophage gene, R2, showing how a series of mutations, all errors in the genetic script, were laid out linearly along the virus DNA. The language was simple and linear, just like a line of text on the written page. The response of the Hungarian physicist Leo Szilard to my Cold Spring Harbor talk on the double helix was less academic. His question was, can you patent it? At one time, Szilard's main source of income had been a patent that he held with Einstein, and he had later tried unsuccessfully to patent with Enrico Fermi the nuclear reactor they built at the University of Chicago in 1942. But then as now, patents were given only for useful inventions, and at the time, no one could conceive of a practical use for DNA. Perhaps then, Szilard suggested, we should copyright it. There remained, however, a single missing piece in the double helical jigsaw puzzle. Our unzipping idea for DNA replication had yet to be experimentally verified. Max Delbruck, for example, was unconvinced. Though he liked the double helix as a model, he worried that unzipping it might generate horrible knots. 5 years later, a former student of Pauling's, Matt Messelson, and the equally bright young phage worker Frank Stahl put to rest such fears when they published the results of a single elegant experiment. They had met in the summer of 1954 at the Marine Biological Laboratory at Woods Hole, Massachusetts, where I was then lecturing, and agreed, over a good many gin martinis, that they should get together to do some science. The result of their collaboration has been described as the most beautiful experiment in biology. They used a centrifugation technique that allowed them to sort molecules according to slight differences in weight. Following a centrifugal spin, heavier molecules end up nearer the bottom of the test tube than lighter ones. Because nitrogen atoms, N, are a component of DNA, and because they exist in two distinct forms, one light and one heavy, Messelson and Stahl were able to tag segments of DNA and thereby track the process of its replication in bacteria. Initially, all the bacteria were raised in a medium containing heavy N, which was thus incorporated in both strands of the DNA. From this culture, they took a sample, transferring it to a medium containing only light N, ensuring that the next round of DNA replication would have to make use of light N. If, as Crick and I had predicted, DNA replication involves unzipping the double helix and copying each strand, the resultant two daughter DNA molecules in the experiment would be hybrids, each consisting of one heavy N strand the template strand derived from the parent molecule, and one light end strand, the one newly fabricated from the new medium. Messelson and Stahl's centrifugation procedure bore out these expectations precisely. They found 3 discrete bands in their centrifuge tubes, with the heavy then light sample halfway between the heavy and light samples. DNA replication works just as our model supposed it would. The biochemical nuts and bolts of DNA replication were being analyzed at around the same time in Arthur Kornberg's laboratory at Washington University in St. Louis. By developing a new cell-free system for DNA synthesis, Kornberg discovered an enzyme, DNA polymerase, that links the DNA components and makes the chemical bonds of the DNA backbone. Kornberg's enzymatic synthesis of DNA was such an unanticipated and important event that he was awarded the 1959 Nobel Prize in Physiology or Medicine, less than 2 years after the key experiments. After his prize was announced, Kornberg was photographed holding a copy of the double helix model I had taken to Cold Spring Harbor in 1953. It was not until 1962 that Francis Crick, Maurice Wilkins, and I were to receive our own Nobel Prize in Physiology or Medicine. 4 years earlier, Rosalind Franklin had died of ovarian cancer at the tragically young age of 37. Before then, Crick had become a close colleague and a real friend of Franklin's. Following the 2 operations that would fail to stem the advance of her cancer, Franklin convalesced with Crick and his wife Odile in Cambridge. It was and remains a long-standing rule of the Nobel Committee never to split a single prize more than 3 ways. Had Franklin lived, the problem would have arisen whether to bestow the award upon her or Maurice Wilkins. The Swedes might have resolved the dilemma by awarding them both the Nobel Prize in Chemistry that year. Instead, it went to Max Perutz and John Kendrew, who had elucidated the 3-dimensional structures of hemoglobin and myoglobin, respectively. The discovery of the double helix sounded the death knell for vitalism. Serious scientists, even those religiously inclined, realized that a complete understanding of life would not require the revelation of new laws of nature. Life was just a matter of physics and chemistry, albeit exquisitely organized physics and chemistry. The immediate task ahead would be to figure out how the DNA-encoded script of life went about its work. How does the molecular machinery of cells read the messages of DNA molecules? As the next chapter will reveal, the unexpected complexity of the reading mechanism led to profound insights into how life first came about.